Hello.
Uh,
How are you?
how are you?
All right. Okay.
Hey,
Yeah.
I guess that's right. Yeah?
Yeah.
Great to see you again.
Okay. So yeah, do you have any updates for us, or?
I have worked a little with inscriptions again, but I guess you're talking about the dates from my life.
Yeah. Yeah.
Actually, the lab where I was working something else, so this week I'm to focus on on the on what we're going on, and I'll have a time from today on. Yeah, I'm trying to at least have 20 hours per week on paper drop this week, I guess.
Okay. So you're going through the papers and looking over, reading them.
Yeah, both papers and also the idea that last week we were talking about to somehow state from a mechanical and also electrical point of view the actual point that what we are beginning of the movement of storm in the developmental procedure. So I'm trying to see if it is possible in current available data, model behavior, and that beginning movement. Of course, it would be better if I find some data for some electrophysiological data or the developmental state, but if I couldn't, I'm trying to try to estimate some processes like to see, for example, how long or how actually we could say how long an impulse could go up to a level that it's some to somehow it's a command for various construction muscles, something like that. This is something I would like to work on. Also, there are some other maybe topics that are somehow pointing in the outline, like the amount of energy needed for the bioenamine that comes from only two planets that is valid for the war, and how it be actually possible to relate the energy levels into the model that would be possible to maybe war for the first.
Yeah.
These are ideas I'm going to maybe this week.
Yeah, that's good. Yeah, so I would just kind of write down those topics and kind of refine them as you go along, and so it's good.
I also checked the draft. I'm not sure if you received the notification of
Yeah, I did get that email. So I've been looking at that document, and so yeah, I haven't looked at it in a couple days, but
okay, for sure.
Yeah. All right, that's great. How are you, Morgan? I don't know if you're just listening in or what. All right, so I guess Dick won't be joining us today, and I don't know about Susan. I don't think she'll be joining us either, but it's okay. Yeah, so that sounds great, Vahid. Keep us posted on that, and we'll keep working on that. Yeah. All right. So the chat, yeah. Jesse said he'll be listening in. That's good. Welcome, Jesse. So let me share my screen. We have some things we can talk about. So one of the things that we've been kind of getting into is talking about morphogenesis in different ways. And this is a series of papers on morphogenesis in tangles and sheets. Some of them are kind of physics-oriented, biophysics-oriented papers, but they kind of go through some of these cutting-edge topics. So let's start with this one. This paper is from arXiv. So this is a new journal that Physics Reviews journals put out. This is Physics Reviews X, or PRX. And this is from their first issue, or their first volume, I guess. And the title of this is Tension Remodeling Regulates Topological Transitions in Epithelial Tissues. It has a kind of a alliteration quality, but basically it's talking about tension remodeling and how that plays a role in regulation of tissues. So I think we talked about this in another earlier meeting where, we talked about the ways in which we can regulate cells in a sheet or in some other way. And of course, in this case, it's tension remodeling. So it's following up on that line of inquiry. So the abstract reads, cell neighbor exchanges play a critical role in regulating tissue fluidity during epithelial morphogenesis. So this is where, cells are kind of next to one another in a sheet, and their neighbors exchange things, sometimes information, sometimes physical signals, sometimes chemical signals. And so these neighbor exchanges play a critical role in regulating tissue fluidity. And when they say tissue fluidity, they mean like the sort of the nature of the tissue, the behavior of the tissue, coordinated migrations, and things like that. And so we've talked about epithelial morphogenesis. Actually, Susan has been on this topic quite a bit. With epithelial cells, they form a sheet, and then they, do there's certain movements that get made in epithelial sheets that are very interesting from a biophysics perspective. So in vivo, which means that they're doing these things within the context of a tissue and an organism, these neighbor exchanges are often hindered by the formation of transiently stable fourfold vertices. So this is something that Susan brought up at the end of last year, and she was talking about this literature on trijunctions. So if you haven't caught up on that, so what happens in a lot of these epithelial sheets is you get different junctions between the cells. So if we have, say, like a sheet of cells, I'll just draw a sheet of cells here. You'll see that there are these junction points. And the junction points will have sort of a fold aspect to them. So in this sheet, we have a threefold junction, or a trijunction. And this is maybe where you have three cells that meet together, and there's a corner that defines that union. Or you could have a quad junction where, four cells come together and they meet at this vertex. We can call it a vertex if we think about this as a network, or if we think about this as sort of an interacting system. And so these different junctions people think that there's some sort of role in maintaining the stability of the tissue, maintaining sort of the dynamics of the tissue, and maintaining the interactions between the neighbors. And so these trijunctions, there's an emerging literature on this, and Susan would be better to speak to about this, but basically this is a literature that started in about 2018 and is kind of a hot topic right now. So they're talking about in this paper these quad junctions or these four-way junctions, and so that's what we'll be talking about here. And so neighbor exchanges are often hindered, in this case, by the formation of transiently stable fourfold vertices. So these fourfold vertices aren't like, they don't exist for very long. As the cells are moving around in the tissue, you get these fourfold vertices that, but they can also hinder sort of this migration because you get stickiness, you get this sort of, these constraints on the cells. And so that's what they're getting at there. So you have these transiently stable fourfold vertices, which can develop into complex multicellular rosettes where 5 or more cell junctions meet. So these are structures that have 5 or more cell junctions. And so despite their importance, the mechanical origin of multicellular rosettes, where you have these density of cell junctions, have remained elusive, and current cellular models lack the ability to explain their formation and maintenance. Here we present a dynamic vertex model of epithelial tissues with strain-dependent tension remodeling and mechanical memory dissipation. We show that an increase in cell junction tension upon contraction and reduction in tension upon extension. So we're talking about these cell junctions. We talk about tension upon contraction. So as the cells contract, they provide tension. And then there's this reduction in tension upon extension. And this can stabilize higher order vertices, temporarily stalling cell arrangements. So this can be like this sort of jamming phase transition that we've talked about in tissue formation and in soft materials, where if you get a certain density of particles or of cells, it can, there can be a flow of them, say through a pipe or through some sort of throughput space. And then as they get more densely packed or packed in a certain way, they can slow down and stop. So that's a jamming phase transition. You go from motility to this sort of stoppage, this sort of quiescence. And so that's what they're kind of getting at here. They're trying to identify some of these phase transitions, some of these mechanisms behind the formation of these higher order vertices. On the one hand, inducing mechanical memory dissipation by relaxation of junction strain and stress. So there's these strains and stresses at each of these junctions, particularly at the trijunctions, but also at these quad junctions, and then the rosettes with their higher order number of junctions as well. Facilitating cell neighbor exchanges. We demonstrate that by tuning the rates of tension, remodeling, and mechanical memory dissipation, we can control topological transitions and tissue material properties, recapitulating complex cellular topologies seen in developing organisms. So they talk about fluidization of epithelial tissues, which is important in a lot of things in early development, cell migration, wound healing, etc. So the ability for epithelial cells to move freely and change their configuration is important in a lot of development and regeneration. And so this is the kind of thing that, they're trying to understand what happens when these things get, when these things stop happening or start happening, or, when they're facilitated or constrained in some way. So that's what they're getting at here. During a neighbor exchange process occurring via a T1 transition, two cells in contact shrink their shared junction to a single point, forming a fourfold vertex. So this is where the cell will, change its shape. They're not necessarily just these squares, but like these two cells will join. Like they'll have this space here, and then as they start to sort of shrink back, they'll come into contact with other cells, forming a junction. So you have to think about this in terms of the cell changing its shape and its size over time. So this happens, dynamically. This fourfold vertex then extends in a new intracellular junction in a direction orthogonal to the contracting junction, while neighbor exchange processes rely on the instability of fourfold vertices. In vivo experiments showed the fourfold vertices can be stable for long times in developing tissues. So during a axis elongation in Drosophila, and these can persist up to 15 to 40 minutes. And we can also see this in other systems as well. So let me see if we have any figures. This will make this a little bit easier. They have some mathematical modeling that they do here. This is their vertex model, attention remodeling. So they model the vertices, and then they also have the tension remodeling as a result. So this is where the cells are remodeled and their junctions are remodeled. So they show kind of the physical model of this. They use a dashpot and spring model to model these interactions between the cells and the formation of these junctions. And they have a phase diagram here where they show the stable and unstable modes of this. So this is a picture, actually. We kind of revisited this kind of a diagram from Susan's work. I think in December she talked about this, where you have these diagrams where you show these junctions in an array of cells. So here we are, this diagram up here, representative section of a simulated epithelial tissue with tension remodeling. So you have these cells and they have these irregular shapes. It's almost like a tiling of some type. And you have these red dots, which are the junctions between the cells. And as you can imagine, as the cells change their shape and their size, and they contract and expand against one another, these junctions change. So you see here you have, I think this is a 7 or 8 fold junction here where I'm pointing my mouse. I think it is an 8-fold junction where you have 8 cells coming together at one point. And so they're all kind of centered around that point. This is, I think, a 6-fold junction. I think this is a 4 or 5-fold junction. And this is a 4-fold junction, and so forth. So Susan was really onto this trifold junction literature. And this paper suggests that there's many more types of fold junctions that we can understand in their role in development. This B here, this figure B, is normalized junction length and tension as a function of time relative to the fourfold vertex creation for instantaneous and delayed T1 events. So this is for T1. This is normalized junction length. This is time relative to fourfold vertex creation. So you can see that there's this, you have larger, I guess the junction length is larger for shorter and longer times. And then there's this decrease in the middle of this time relative to fourfold vertex creation in minutes. So you get this sort of pattern where, we have cells that contract against one another, form a junction, and then they expand maybe against one another and the junction goes away. So just showing these dynamics. And so in this figure 3, tension remodeling promotes asymmetric length distribution. This is a histogram of junctions at steady junction length at steady state. So this is remodeling versus no remodeling. We see the fraction of total junctions on the y-axis and the normalized junction length here. So this is normalized junction length as a function of fraction of total junctions. So in other words, with no remodeling, you get these normalized junction lengths of 1, which are most common. And that's a unimodal distribution from 0 to about 1.75 or so. With remodeling, you get a wider variety of different junction lengths. And as we might expect, each of those are a smaller fraction of total junctions. So you have this broad spectrum from like 0 to almost 3 in terms of normalized junction length. So this is just basically saying that remodeling induces a lot more variety in the junction length. So this is just means that cells are moving around a lot more, and they're more dynamic, and they're making these different types of connections, and they're moving against one another. Whereas with no remodeling, you get these sort of jamming states where they're kind of stuck in a mode, and so that's basically showing that. Then this is actually where we have in B, correlation between total junction tension and junction length in tissues with blue and without black junction remodeling. So this is without junction remodeling and with junction remodeling. So blue as with junction remodeling, you get a wider variety of junction lengths. Normalized junction tension increases as the junction length decreases. The same isn't necessarily true where you have without junction remodeling. So when the junctions don't remodel, you get sort of this narrower range of junction lengths, and the junction tension is more or less constant. So yeah, and then that's basically support for A, that there are these dynamic things going on in the tissue. And so this is interesting here. This figure, this is frequency versus the number of sides. So this figure talks about tension remodeling influencing the geometry of cell packing. This is where we have solids versus fluids, or a solid regime and a fluid regime. And we can see that there are these different shapes that form. So this figure here, C, this was like something that Susan was interested in terms of trijunctions. These are where you get these triangles, these octagons, and these nonagons. And they're just different shapes that form based on the junctions and the cells around them. And so you see that in a solid, these shapes are basically all sort of a frequency of almost 0. In a fluid, they jump up, or at least the octagons jump up greatly. Triangles increase with the KL proportion here as that increases, and the nonagons don't really increase that much, but they do a little bit. So there is a greater diversity of sort of different types of junctions in fluids. Octagons become more common in fluids. In this case, we have a solid fluid transition. These are squares in red, pentagons in blue, heptagons in purple, and hexagons in green. We can see that hexagons are dominant in solids, but that once you get to a fluid, the number of hexagons falls and the number of pentagons increases, and the number of squares increases as well. And heptagons are always kind of rare, but they're, kind of a constant between solids and fluids. So you can get from these figures that there are differences between solids and fluids in terms of these junctions and the shapes they form. And it's just evidence, again, that, when cells move around in different media, in different contexts, that they have these different properties. So this is where we look at the geometry of cell packing. So we see that this is a graph showing the number of sides versus the frequency. So you see these different K, KL values, and you end up with these different shapes. So we have this shape here, which is dominant, and we have other shapes. So we have a pentagon, a hexagon, and so forth. And so we can see that there's a hexagon. Hexagonal arrangement is the most common, and the triangular is the least common. And so I guess this is the nonagon down here. And so we can see this unimodal distribution centered on the hexagons. So that's enough about that paper. I thought that was interesting in light of some of the things we've talked about with respect to that. This is an interesting paper from the arXiv. I'm not sure what journal this is published in. It might not be published yet. But this is from a group that includes Albert Laszlo Barabasi, who's, of course, a big networks person. And this is a paper on bundling by volume exclusion in non-equilibrium spaghetti. So this is not directly doing biological experiments or looking at biophysics, but it talks about physical networks, and the examples they give here in the abstract are like the brain or metamaterials. So the abstract of this paper reads, in physical networks like the brain or metamaterials, we often observe local bundles corresponding to locally aligned link configurations. Here we introduce a minimal model for bundle formation. You'll see this down here below. Modeling physical networks as non-equilibrium packings of hardcore 3D elongated links. And so this is where we have these, again, these packings that shift and move around. And so they model these as hardcore 3D elongated links. So they're elastic, but they have this 3D property. We show that growth is logarithmic in time, in stark contrast with the algebraic behavior of lower dimensional random packing models. Equally important, we find that the slow kinetics is metastable, allowing us to analytically predict an algebraic growth due to the spontaneous formation of bundles. And so this offers a mechanism for bundle formation resulting purely from volume exclusion and provide a benchmark for bundling activation and growth. So they're interested in the assembly of physical networks that have these kind of properties. So these include neural tracts, which are bundles of nerve fibers, biopolymers, and so these are different types of graphs. So when we think about graphs, we think about the standard type of network that we're, usually use for modeling, gene-gene interactions or protein-protein interactions. This is a little bit different from that standard graph. And so one of the reasons for this is because we're kind of modeling the physics of something. So we're modeling three-dimensional objects instead of like abstracting, information about interactions between things that aren't really geometric in nature. So they have this sort of spatial property, they have this geometric property, and this is one of the things we've been working on with embryo networks, that, there are these, you can extract these networks that describe interactions, but you also have this physical aspect where you have the 3D morphology that you have to characterize, and then there's interactions between the 3D morphology and the interactions and that map. So this is an interesting, and I think Susan did another paper, she presented on another paper where they had something like this, where they were doing, it's like they did a set of experiments where they had like these rods and these packings, and I can't remember the exact paper, but this is interesting in light of that paper, or that discussion. So, the impact of volume exclusion has been widely explored in the realm of equilibrium granular and colloid materials made of short rod-like particles. So this is an example of a rod. And these rods, you see them all the time in biology. You see different types of bacteria have rod-like shaped morphologies. You have other types of like nerve bundles or nerve fibers that have this kind of morphology. So it's common in biology to see this. Little is known about its influence during the non-equilibrium assembly of elongated filaments, like those characterizing the architecture of physical networks. And so it's unknown whether volume exclusion alone can give rise to link bundles in a system far from equilibrium, as existing models either assume cross-link interactions or operate at equilibrium conditions. So this is their model of sort of one of these bundle networks. These are bipartite 3D links. So we have three-dimensional links, and we have this bipartite network. And so this rod here, or this rod shape, this actually shows a deposited link. So you have this node, and then you have this halo excluded volume. So this is like a volume that follows this link downward from this surface down towards. So we have z equals 0 and z equals 1. The node is at z equals 1, and there's this halo excluded volume that moves down to z equals 0. So you have this initial position at z equals 0, x, y, and you have this excluded volume that comes from the next time point, or I guess the next position in the z dimension that is kind of linked to that. It's going around. There's a radius of that. So in other words, if you have a rod, you can measure it from sort of its base, and it has this sort of angular gyration radius. And then you can measure the excluded volume from the base to the object that's going around this volume. So this whole volume can be excluded, and so you end up with a model like this, where you get these rods or these excluded volumes that cut across one another, but they form this dense volume here. And so this is the way they build their network. So basically, they're taking the idea of nodes, they're building in a volumetric aspect to it, and then they're using that as part of their network. So keeping only the essential properties of these systems, we introduce a bipartite model consisting of physical links modeled as slanted cylinders of diameter, whose endpoints are constrained to the opposite faces of a unitary cube. So the endpoints are constrained to the faces of the cube, and then they go within the cube. We deposit the links by random sequential deposition, and it's just the method that they use. We show that this physical link model can be solved analytically, enabling an exact comparison against simulations. We find that at the temporal onset of physicality, the packing kinetics undergo a dynamical transition from a non-interacting regime to a strongly interacting one, where the density of links evolves logarithmically in time, in stark contrast to the algebraic growth observed in 1 and 2 needle-like systems. This is in citations 41 and 43. So 41 and 43 are down here. So we have this paper from 1991, asymptotic results for the random sequential addition of unoriented objects. And this paper 43, from car parking to protein absorption, an overview of sequential absorption processes. This is from 2000. So this is kind of following up on a problem that's been around for a while in soft materials. So these are the kinetic instabilities. So you see that they have their volume here. This is their network. And they try this they do a nearly saturated packing of links and for different parameter values. And you see that there's a difference here. So they test this for different parameter values. And then they show bundling formation and bundling onset. So they actually can show these things happening in the network as a consequence of the way the network is structured. You see a lot of local bundling, which is this picture down here. And, there's a lot of things you can get out of this kind of model. Okay, so that's that paper. The next one here is inflation collapse dynamics, which is, of course, something that we see. This is maybe more biological. This, I think, is done in organoid cultures. So this is something that Morgan might be interested in terms of like how some of these morphologies happen to form. So they use this inflation collapse dynamics model. And so let's get into it. Let's look at the graphical abstract first. This is where they have these triple-decker sandwich cultures where they can do, they can facilitate uniform growth. The organoids that they grow have these stem cell zones, characterized by expression of LGR5. So these are LGR5 positive stem cell zones. Then you get this inflation collapse driven fission mediated by piezo-1, which is a pressure sensing protein. So you get this mediated inflation where you get the zone that inflates in size, and then you get increased pressure in this zone. And this is where piezo-1 is mediating LGR5 loss. So as the zone increases in size, the stem cell zone, it starts, the pressure of that zone starts to shut down the expression of LGR5. So it's just like if you're blowing up a balloon and you get a higher pressure of air inside the balloon, if something were in the balloon that's pressure sensitive, it would be affected. And so this is kind of what is going on here. And then you have this inflation collapse driven fission mediated by piezo-1, where there's budding during the collapse. So this stem cell zone, the cells shut down of their expression of LGR5, and then that large zone collapses into these buds. So you see these buds, which are now all LGR5 positive, that start to happen during the collapse. And then you have this BASP1 clue positive stretch state, where you get the stretching of the organoid and these buds that kind of come off the edge. And so they change shape, and you see these areas here in red. And then inhibit CFTR or piezo-1. So if you inhibit those proteins, you get fewer fission events. So you don't get as many fission events if you inhibit certain genes and proteins within this system. So this is just kind of looking at how organoids grow and change shape, and how they get these stem cell zones where they can then do further differentiation. So in brief, intestinal organoids contain stem cell zones that resemble the crypt base in vivo. So they're stem cell crypts in organisms, where you have stem cells, sources of stem cells that come out, and we have them in the brain, we have them in other areas of the body where they replenish cells. It provides an engine for regeneration. And so in this case, this is what organoids are doing. We studied SCZ patterning by live imaging and perturbing organoids in sandwich cultures, which are these things up here. We found that SCZs undergo piezo-dependent fission, driven by inflation and collapse of the organoid lumen. So this is the lumen here. And inflation induces a stretch-associated cell state. So when you inflate it, there's a stretch-induced cell state, which is the shutdown of this LGR5 marker. And so you can actually change cell state with pressure, basically, is what they're saying. And the pressure of inflation of the surrounding tissue. So this kind of goes through some of this, some of the dynamics of fission and some of these biophysical states that we observe in these systems. And so with one of the important things about organoids, and actually about tissue formation in development and in cellular reprogramming, is you need niches, which are these spaces in the organ or in the tissue or in the culture that allow for the differentiation of certain cells, sometimes to maintain stemness. But we get these niches in this, so that's what they're trying to recapitulate. So let's see. The intestinal epithelium is a stereotypical self-renewing tissue. So they're trying to model the intestinal epithelium. This is a model that allows us to study self-organization and niche control. In most mammals, the intestinal epithelium undergoes a near-complete turnover every 3 to 5 days. Supporting this turnover, equipotent LGR5 positive intestinal stem cells. So these are ISCs, and this is where we're getting our source of intestinal cells, and intestinal epithelial cells, which cluster in stem cell zones at the bases of invaginated crypts. And so then ISCs differentiate into several cell types that emerge from crypts in villi and form the luminal surface of the intestine. In homeostasis, which is what we're, in a tissue, you would see the sort of homeostatic mechanism. Cell division at the crypt base precisely balances cell loss at the villus tip. So these crypts that they show model the villus tip. So they're modeling different aspects of the in vivo tissue. An extrinsic niche formed by the underlying non-epithelial mesenchyme supports these behaviors. As cells migrate up the crypt-villus axis, they lose mitogenic stimulation and become exposed to signals that promote differentiation. So basically, this aspect of the if we go up to this figure again, we see these structures here, and they're modeling the villi basically. And so this is what they're trying to sort of recapitulate. And they're sort of doing it. It's not perfect, but that's what they're trying to do. And the argument that they're making is that if you're in a real tissue, there is this aspect of there's a micro environment or a niche that enables a lot of this to happen. And so they're able to sort of recapitulate this niche through this, these sorts of crypts or these pressure things. So this is something that is I think it's an interesting paper from the standpoint of, how realistic are organoid models with respect to actual tissues. And what can they tell us about a lot of the processes going on in those tissues? So the final paper in this group is creative processes during vertebrate organ morphogenesis, biophysical self-organization at the supracellular scale. And this is from a group including Amy Shire and Alan Rodriguez. And we've talked about some of their work in meetings last year. We were talking about some of their work on epithelial cells, in fact. And, they're interested in self-organization and morphogenesis. So this is an interesting paper that kind of draws out that idea further. So this is what they talk about as creative processes during morphogenesis. So what they mean by creative processes, let's see what they mean. So here we review recent developments in the literature that provide insight into self-organization at supracellular scales in vertebrate organ morphogenesis. So we sort of saw, examples of tissues, specific tissue types, and models of those epithelial tissue types in particular. What they're looking at is supracellular scales, which could be, organs or it could be tissues, mostly organ in this case. We briefly present a historical and conceptual analysis of the term self-organization. Based on this analysis, we suggest that self-organizing processes, at their root, possess a form of causal relationship, reciprocal causality. And this is, I know that Jesse and Morgan and I were at our meeting on Saturday. We were talking about cybernetics and some of the things, that they think about reciprocal causality a lot. So it's kind of an interesting point that this comes up here. So this reciprocal causality is markedly distinct from linear causal chains. We survey the extent to which reciprocal causality can be used to interpret or clarify supracellular studies in development and disease. Finally, we explore how reciprocal causality can exist across length scales. Identifying situations or multiple scales require simultaneous analysis. So this is an interesting paper on sort of some of these ideas about feedback and tissue morphogenesis and even organ morphogenesis. So this is their model system that they use experimentally. They do this stuff with feather follicle formation. So they're interested in sort of the patterns that form in feathers and birds, and I think in chicks what they're doing. So this shows an example of how this process works. So you have this day 6 embryo with an anterior-posterior orientation, and over 2 days to day 8, you have this pattern of feather follicles. So there are these little buds on the surface of the embryo here, oriented in this line from anterior to posterior. But you notice that there's this pattern where they're sort of distributed in a certain way. And so this is because these processes are sort of spatially restricted. You have these cells here. So, before follicle initiation, skin is a uniform bilayer consisting of epithelial and mesenchymal cell layers. So up here you see the epithelial cell layer at the top. There are these square cells arranged in a single layer. And then this mesenchymal layer, which is an orange. So you get, and so then you get this sort of this aggregation of these cells here. They push up, these mesenchyme cells, they push up on the epithelial layer and they form these buds, which you see here. This is from day 6 to day 8. So you have this disorganized set of cells on the bottom layer. They kind of bunch up. They start to push up on the epithelial cells on the top layer, and that deformation leads to these buds, which will eventually end up forming these feathers that will come out. It's kind of like hair follicles. And so you get these, basically what you have to do is you have to get these aggregates. So these cells have to aggregate in some way. And the aggregates have to be patterned. They can't just be, in random positions. I guess they could be I guess you could say they're quasi-random, but they have to be in a way that covers the organism. The feathers have to be organized. And as we see with hair, with feathers, there's this spatial organization that's important. So they show this model here in C of substrate resistance versus cell traction. So these cells have to have some type of resistance against the substrate. So they have to be, pulled back by the resistance of the substrate, but they also have to have traction to push them forward. And so you see there's this reduction in resistance and this increase in traction over time or over these different conditions, which is that in the no aggregate condition, you have a lot of substrate resistance. It's almost dominating the system. When you have many aggregates, you have a mix of substrate resistance and cell traction. And then for when cell traction dominates, you get one aggregate, which is also not desirable because then you would end up with a single large bud here in this layer, and you'd only get like one very large follicle for a feather. So that wouldn't be good either. So it's a balance between these two to get these many aggregates. And so that's the problem they're trying to solve. They point out that there is no molecular blueprint instructing each cell to decide to join an aggregate. Conversely, the spontaneous formation of supracellular aggregates informs expression levels of various signaling molecules. And so in other words, new information and structure are created by self-organization at the supracellular scale. Self-organization matters for vertebrate organ morphogenesis because it provides a means for growing tissues and organs to increase their complexity and create new ordered forms. It is a fundamentally creative process whereby new information and structure are generated. So what they mean by creative process is that there's this new information and structure that you see here. You go from disorder here to order, and it isn't just about like the up and down regulation of genes. There is this biophysical part of this, which is important to get this also regulated in a way that you can get, say, you don't go too far. It's sort of a happy medium is pattern formation is. You have this sort of totally random state, this totally clustered state, and then it's something in between that. And so that is what they mean by creative process, that regulation ends up in this creative process that creates this kind of order that is, beneficial to the organism, or at least beneficial to the organisms we see out there today. And so they talk about this sort of the key features of self-organization here. They throw out a lot of terms in this. And so they talk about actually what people have talked about in books. So in a lot of the literature, there are a couple of features of self-organization that people kind of focus on. The first is that biological systems comprise a hierarchy of length scales. So, you start from like the small scale of interactions between cells to tissues to organs, or, you have like these local interactions or these global interactions. And this forms a hierarchy, so it's a functional hierarchy. If you think about a tissue, it has subcomponents and so forth. 2 is at the lowest scale in a given system. Independent entities interact locally. So there's a lot of local interaction, as we mentioned in the other papers. 3 is that complex structures or patterns show up seemingly spontaneously at higher scales due to the interactions at the lowest scale. So cells that are interacting, like we saw in the first paper, they form these junctions and they do things, large scale things like heal wounds or form structures and development. And then 4, structure pattern formation is not coordinated in a centralized way. There's no pre-existing mold or template. So that's from these citations. Collective cell migration exemplifies these features. Cell-cell and cell-substrate adhesion, repulsion, polarity alignment, and other local interactions give rise to collective cell movements with strikingly large-scale structures such as flocks and fingers. We kind of saw that with the organoids where they're forming, even outside the tissue, they're able to recapitulate that micro-environmental niche where, they can kind of do similar things to what's happening in the intestine in a culture. So this is interesting, the way that this kind of is defined. They actually propose a 5th key feature of self-organizing systems, which is that they require an external energy source. If the energy source is removed, self-organized order decays. So this is, again, with respect to open systems in biology, but also elsewhere, the idea that a closed system will degenerate, an open system will remain viable. And so this is a little bit harder to understand in terms of development, but you have to think about maybe like an embryo, maybe like a seed or an embryo that doesn't have water or energy or something like that. It doesn't develop. So these are kind of, things that are mentioned in the literature. And then, they get into this idea of reciprocal causality. So that means that when you have causality in one direction, that's not enough. They talk about this as kind of a first principle in development. And so they basically talk about molecular signals and collective cell mechanics as being one mechanism for this reciprocal causality. So you get molecular signals that are acting and cell mechanics that are acting to sort of mediate those molecular signals, as we saw in the organoid example and in the other examples as well. These tissue geometries establish local differences in cell packing and morphology, which associate with local differences in some protein concentration and activation and cell fate. So this is, again, a set of things that sort of interact with one another and form this reciprocal loop. Reciprocal causality need not involve specific molecules as causal factors, but can instead involve simultaneous mechanical exchanges between 2 material entities. Analogous to liquid crystals, mouse myoblasts under circular confinement move in a line in such a way that they exhibit a single topological defect, resulting in a spiral configuration at low density or an aster configuration at a higher density. So this is, again, going back to this literature on liquid crystals and soft materials. So this is really kind of a nice paper. I don't want to go through the whole thing. We're kind of at the end of our time for this hour, but I just wanted to present those 4 papers that kind of present a set of ideas and kind of, kind of feeds into some of the things we've been talking about with biophysics but also with self-organization and systems. So I don't know if we want to have a discussion about that or if we have thoughts. Yeah.
Well, thank you for your presentation. Actually, it's not my field of expertise, but I've been in contact with Susan. If there is anything related to computational simulations that would need coding, I would be glad to participate. And if you are also working on some related biophysical topics, something like this, I would be glad to participate on especially on the programming part, and somehow on the actually I would be maybe could be participate and add something to for the programming part, but would be interested in learning something new for the biomechanical and biophysical simulation to add something to my knowledge as well. So I would be glad if there is any ongoing work to participate.
Yeah, that's great. Yeah, I'll keep you posted on that. Yeah.
For sure. Thanks.
So I know we're a bit limited because Susan usually has like a lot to say about this kind of papers, but
yeah, I've been once in contact with her, and we're talking about how she's actually working on her thesis, and yeah, I guess it could be really interesting for her if she were available. I guess she's on holiday, maybe, or something like that. She said.
Yeah. Yeah.
I could come up with some short updates for my work for the next week. So if you're interested.
Yeah, I think that'd be great. Yeah.
For sure.
Morgan or Jesse, did you have anything to add? Yeah, that's great. Yeah, thank you for attending the meeting. So yeah, if we have any other questions, let me know, and we'll talk next week. Thank you. Have a good week.
Thank you.
Yeah.
Goodbye.
Bye. Now I'd like to follow up on some ideas in the last paper about reciprocal causality. I want to interpret that in terms of maybe like classical cybernetics, and in terms of feedback loops, and in terms of what that might look like in a cell or in a tissue. So let's start out with sort of a classical first order feedback closed loop cybernetic system. So we start with our input. And our input is usually something that we're inputting to the cell. So we think about like maybe we talked about biophysical constraints or like pressure input, pressure sensation of the cell. The cell encounters in its environment. So that input goes to this controller. And this controller is basically going to integrate things in the system. And you'll have this first order feedback loop that tells the controller what needs to change based on the system. And then the system state has an output. So our controller in this case is the cell state. So for example, in a stem cell experiment, I have a stem cell which is in a certain state, and I want to do things to make the cell differentiate. So I might deliver some sort of pressure cues, I might deliver some sort of viral vector that allows the cell to reprogram. There are a number of inputs that I can put into the cell state that will change the state. But it isn't necessarily changed at this first point, which is the controller. The controller is just merely taking signals from the input and then passing it to the system, which might be like a gene network. And then that gene network there's some effect there that gives a feedback signal to the cell state. So this is feedback, this is feed forward. Cell state basically is taking this information from the environment, passing it on to the gene network as a feed forward signal, and then getting feedback from the gene network after the effect has happened, and then that determines what the effect is on the cell state. And eventually we can get enough of this feed forward signal so that the gene networks, whatever changes are happening in the gene network, feed back to the cell, and the cell can change its state. So we can measure the cell state at C, at the state of the controller, or we can measure the gene network state of the state of the system, independent of feedback. But of course, it's not really independent of feedback, because the feedback will tell the cell state, what it should be, and the contributions of this input, the contributions of this feedback will provide us with an indicator of the cell state. And then there's an additional feed forward signal from the cell state that affects subsequent iterations of the gene network. So the gene network and the cell state are tied. The environmental input is tied to the endogenous activity of this gene network, and then that provides feedback. And so there's this reciprocal causality. And that is the contributions of this feed forward signal and this feedback signal that gives us a final output signal, which is the integration of cell state and the gene network. And we can do this over time, so we can measure this as sort of a dynamical system or a sort of a time series, so a T, 0, 1, n, meaning that we can measure it at any point in time, and the thing will evolve according to what's going on in the network. If we think about those contributions, we have our feed forward signal and our feedback signal. Now what's interesting here is that the feed forward signal, it could be something like pressure. It could be something like, what we talked about, these cell junctions, these trijunctions or these fourfold junctions or these eightfold junctions. And one of the things that it's doing there is it's exchanging things. So we could say cell exchange. And of course, as we know, in that case, those cell exchange parameters are always changing. So we can do, we can basically have a variety of feed-forward signals that are affecting the cell state, but also affecting that gene network, so it can provide feedback to the cell state. So it's correcting the cell state. I can introduce pressure to a stem cell. It might remain a stem cell until it gets a feedback signal from the gene network that will provide some sort of instructive cue to differentiate. Now, the way of looking at that, of course, is purely in terms of feedback as a consequence of the feed-forward signal. But you can also have endogenous feedback from the gene network. So we could have, endogenous activity, which usually means like that there's some activity, some ground state of the gene network, where it's on or it's off. We can also have noise. And noise can sometimes facilitate things in systems. But also you can have these signals, and they sort of guide, they maybe turn on and turn off certain genes. So we know that they're, we often use in experiments, we use markers. So something is piezo positive, piezo1 positive, for example. That means that gene for that protein is being expressed, and we can actually get, use that as an indicator of whether there's significant feedback or not. So, marker positive expression. And again, it's not so much that that marker is responsible for what's going on. It's that it serves as a way to, it is an indicator as to whether or not that signal is actually feedback. So we have feedback, which can be endogenous activity, marker positive expression. We have feed forward, which are these environmental cues. And the idea here is that these environmental cues, can say, and let me do this. So we can have like an input, and this is actually a feed forward to the cell state. So our cell state basically, if we say, if our cell encounters something in the environment, it's probably not sufficient for that environmental cue to change the cell state just by encountering the cell. So in other words, if the cell encounters pressure, a pressure gradient, it's not going to change its state just on its own. It's going to have to involve a number of things. And there are a lot of things we could have in the system. I'm just simplifying this for the case of demonstration. We have this gene network. And there's another feed-forward signal here. So the feed-forward signal from, say, the environment to the cell state exists, but it's not enough alone to trigger any sort of change. We also have this feed-forward signal from the environment to the gene network, but that in itself is not enough to change the system necessarily. Well, what may change the system is this first-order feedback. And when I say first-order feedback, I mean that there's this immediate loop between this feed-forward signal from cell state to gene network and then feedback, which is what the state of the gene network is after it's encountered the signal. So, a lot of times if you have something like pressure or something like, crowding, or I think there are other chemical cues that we might consider to be correlates of biophysics, or, cell-cell communication, you often get signals that are transducted and converted into signals that trigger genes in their expression. So that's what I mean by the signal being a feed forward to the gene network. The effects of that then have a feedback on the cell state. So there's a causality here between environment and activity, and then a reciprocal causality between activity and state. Okay. And so one could misinterpret that as sort of the causality of environment and state. If one were to look at it very simply and say, well, look, if I poke this cell with an environmental stimulus, it changes its state. So that must mean that the environmental stimulus is changing its state. Well, that's not as simple as it is. It's not as simple as that, because you're actually triggering things within this network. And you have this reciprocal causality between maybe an environmental signal. And you could do this experimentally by testing, a gradient of environmental signals, or testing it and seeing, how often the environmental signal triggers some change in the gene network, which then triggers a change in the cell state. So, thinking about it in this way in terms of reciprocal causality in terms of a system is much more useful than thinking about it just in terms of a single cause and effect relationship. And so this is something that, and again, one of the problems in biology is that, we have really problems of context. It's like understanding what the context is of some stimulus and what the response is. This is the same thing with behaviors that emerge from the brain. It's hard to really kind of get a handle on simple cause and effect relationships. And this is maybe one reason why, because we have this reciprocal causality. Now, what's a more fun exercise is to think of the system not just as a single entity, but to think of this as like a very wide-ranging network. So we can think of we can have our controller, which is here, which is cell state, and we have all these different gene networks, which are systems. So we have these different systems that are affected by, or maybe are part of cell state. And so we have all these systems that exist out here. And we have all these feed-forward signals going from environment to cell state to these systems. And so, when we say that cell state is passing on a signal, we just mean that its reference point is at time 1. So I can take this input signal that feed forwards to the cell state. At time 1, the cell state is a stem cell. And then at time 2, that feed forward signal is relayed to the gene network. And then at time 3, there's a feedback from the gene network back to the cell state. And then at time 3, if there's a change there, then that's what we record. So we actually have this time series where we're measuring the system, the controller, at discrete states. We're getting the signal integrated either through simple causality or reciprocal causality, and then that's giving us, a measurement at, say, like time 3, and then whatever subsequent effects that feed-forward signal has, then that's captured in later time periods. But it's not as simple as that, because we have all these feedback signals that are a consequence of getting like a communication relay back from the gene expression or the gene network to the cell state. So all these feedbacks are of different orders. These are all first order feedbacks. But if you have say like a relay of gene networks, you can have things like second and third order feedbacks, where there's a distance between the relay of feed-forward signals and the feedback signal. So if I have a feed-forward signal that goes, say, which is a first-order feed-forward, that goes to this gene network. That's just a simple signal that gets relayed forward. But imagine that this gene network then has something that happens to it. It transforms this information, and then it passes that transformed information onto another gene network. And then this happens again. So this information is pretty transformed, and it's in a different kind of mode. It may be that the signal is, embedded in noise. It could be that the signal is refined. But we have this transformation of signal, this feed-forward signal. Then if we give a feedback signal back to the controller, this is a very different feedback signal than the feed-forward that's coming in from the environment. So we can actually get this is called a third-order feedback. And this third order is much different than maybe like a first-order feedback. Because the first order feedback might be like a simple difference. So we might say that this transformation here, actually this feed forward to this gene network here, gives us a first order feedback, which is maybe like the difference between the original feed forward signal and the feedback signal. But in a third order system, it's a lot more complicated. It could be like a very complicated mathematical transform. It could be a very convoluted signal that gets fed back, but it is feedback. You can think about this in terms of if you have a circle of people and you start a story off in one person at the head of the circle, and they pass the story on to people around the circle sequentially. And the story will make it back to the original person who told the story. But at every step, that story is mutated in some way so that it's changed by the retelling. And so by the time it gets back to the original teller, it may or may not be unintelligible. It verges on unintelligible. And this is sort of the thing with third order feedback or higher order feedback, is that it's going to be maybe unintelligible. Maybe it's embedded in a lot of noise. Maybe it's not something that looks anything like the original feed-forward signal. But this is something that still happens in a lot of systems, and something that is actually somewhat useful to the system as a whole. It's something like cell state, a controller, which basically tells, maybe it provides robustness, maybe it tells the cell additional information about what's happening downstream, and so forth. So there are a lot of things that are going on here. I wanted to present a little bit of a sort of set of ideas about reciprocal causality, feed-forwards, feedbacks, and their role in biological systems and complex biological systems. And so, the final word on this is that if you think about a system that gets built up in terms of complexity, oftentimes if you're building from like a cell to a tissue to an organ, first of all, you're reusing parts. So a lot of these networks are modular, and a lot of these sets of feedback loops and feed-forward motifs are modular, so they're going to be repeated across, say, like cells and tissues and even organs. The second word here is that you do have these complex cases that I showed later in this talk are more common than not. So this first order feedback example is a toy model. It doesn't really give you any biological insight. It may be a very simple system, but in these cases it's not as simple. And so you see the proliferation of complexity here. And you see the variety of feedback signals from a single feed-forward signal. So you can have, multiple copies of that feed-forward information go to different places, and then the controller gets feedback from all these different places, a diversity of feedbacks, both in terms of where they come from, what the transformations were, and then this ordering effect, where you get this third-order message that's very different from the kind of feedback you would get in a first-order message. And so these are the kinds of things, and then the reciprocal causality part comes from the notion that, feedback is just as important as the feed forward, that the feedback is kind of pulling back on the feed forward, it's kind of correcting it, or sometimes it's contradicting it, but maybe in a way that regulates the system in a certain direction. So this reciprocal causality is kind of hard to really see. I didn't give any really good examples, but if you look at like any paper on different types, they'll do experiments on a gene, what they'll call a gene circuit or a set of candidate genes. They'll say, this thing is upregulated, and then downregulates some other gene, and then upregulates something else. And you see this in brain activity too. The different brain regions or neurons can inhibit or pass on an excitation signal. In fact, a lot of times in neuronal networks, you have this festival of inhibition, where the inhibition, it's always about the cell, the output being an inhibition signal to the next cell, and that's how you kind of gain control. So the feedback is actually inhibiting the feed-forward signal. And so that's the causality. That's the reciprocal causality. The information is going outward to some entity that's then returning a signal that's causing it to sort of pull back. And so there are a lot of examples of this in like biologically oriented cybernetics. One famous example is rein control, and it's like driving a sleigh where you have two reins and you have to pull on one side and then pull on the other, and you have to do this in a certain sequence or rhythm to really get full control over the system. So there are a lot of different metaphors we can use to sort of describe control and describe this reciprocal causality. But that's all I'm going to say about that. I think the quest for causality, people get hung up on this question, and it's usually the way they think about it is that it's one way. It's like this thing causes that thing. What I'm showing you here is that actually reciprocal causality is really about one thing causing something else, but then being sort of moderated by something, by that thing that's being affected, feeding back to that original thing. So thank you for paying attention, and I hope you learned something.